and her talk today is about the chemical toxicology of endogenously generated reactive oxygen species and their role in cancer etiology. Welcome again, Dr. Amanda, and you have the
Thank you very much. It's very nice to be introduced by a member of the family, so to say. So I really appreciate that introduction. So today I'm going to give you a little short introduction to what we actually do in my laboratory. My title actually talks about chemical toxicology of endogenously generated species, reactive oxygen species. But I'm going to talk a little bit about also radiation-related reactive oxygen species as well. So this is my always introduction slide because I have to get people to remember some of these structures. What we're talking about when we're talking about reactive oxygen species are all of these small, highly reactive species that are generated. On this side, what you see are ones that happen to come from the fact that we breathe oxygen, of course, and our body has to metabolize it. So you see hydrogen peroxide here, you see singlet oxygen, you see the hydroxyl radical, which is, of course, the major oxidant, and then you see down here the oxygen radical. What you see over here is hypochlorite. Hypochlorite is, of course, coming from chronic inflammation, and I will talk a little bit more about that in a second. Over here on this side, you also see nitrogen-based reactive species, and these are also usually related to inflammation. So the endogenous sources are listed in this box: mitochondria, peroxisomes, cytochrome P450, all of those types of enzymes. The exogenous sources, which actually create then reactive species in the body, are here. That's like ultraviolet light, ionizing radiation, chemotherapeutic agents, of course. Inflammatory cytokines are internal, should be over here, and environmental toxins. So if we look down a little further here, we see a barrier. We know then, though, that these species are able to interact with all of the biological molecules, but we list here only the proteins, the lipids, and the DNA. We know that RNA is also the sister to DNA, and we also know that carbohydrates are there. So all of these types of biological molecules can actually be damaged by these species. But my focus in my laboratory has to do with DNA and RNA. And today I'm only going to talk about DNA damage. So when these species react with DNA, what can happen? We can get DNA adducts. Of course, those being species that are modified in a very big way, usually making bulky pieces of DNA that have to be repaired. We can get DNA-DNA or DNA-protein cross-links. Those species are also highly mutagenic. We can get strand breaks. Those create a very difficult situation for the cell. They have to be repaired, and the repair strand breaks is the most difficult situation. And then we get a basic site formation where we simply lose the genetic information by losing the base. So that's a very important aspect as well. And almost all damage leads to some formation of a basic site. So when we talk about the chemical biology of chronic inflammation, it's all about radical chemistries. And as you can see here, this is a little fuzzy, but you can see the macrophages here, T lymphocytes, the neutrophils. These three types of cells are actually interacting here to create lots of different types of reactive oxygen species and reactive nitrogen species. And so then you can see the different components that we talked about over here. We see the hydroxyl radical there, and then it always, of course, affects these molecules. So chronic inflammation as a cause of cancer is now well established. So patients who have Crohn's disease or the other gastrointestinal disorders such as ulcerative colitis, those patients are at very, very increased risk of developing cancer. We also know because of the chronic inflammation caused from ulcers and the bacteria that causes ulcers, that also causes stomach cancers. So those two situations have given us a lot of information about chronic inflammation and its relationship to cancer. So one thing that you'll notice, and everybody knows this, is that DNA usually in the cell is in double helical form and it's B form. So we have, excuse me, the major groove and the minor groove. So is a representation of B-form DNA, and these spots on the outside show us the solid accessible surface area. So we're looking at where actually small molecules like the hydroxyl radical, where they can attack the DNA. So what you look at here, we have color-coded, are the different hydrogen atoms on the sugar. So the research in my laboratory does not deal with the base damage. The nucleobases are damaged constantly. The repair mechanisms for those, though, are very good usually. But the sugars are the place where we usually see the strand breaks occurring. So that's why we decided to focus on this position in the DNA. So you see here there are two hydrogen atoms, of course, at the 5 position of deoxyribose. These two are represented here. So extraction of either one of those by a hydroxyl radical creates a water molecule and a radical at this position. Here is the 4' position. It's seen here. This is, of course, the minor groove. So those 2 hydrogen atoms are in the minor groove. Then we have this yellow one here is the 3'. It's in the major groove. And one of the 5' is in the major groove. We can't see the 2' in this kind of representation because it's buried. So remember the 3', it's in the major groove, and this is the focus of the research in my laboratory in this structural type of DNA. So what happens when you oxidize the sugar? So basically I show here a radical in the 2-deoxyribose that can appear at any one of the 5 carbons because we know that they're all accessible to some extent. Now, most of the time the cell has, of course, hydrogen donation species or some reductant. And so glutathione is the body's most important reductant in this respect. And if it reacts with the radical, then you just actually add the hydrogen back on. So by adding the hydrogen back on, the DNA is perfect and life goes on. However, there are 3 positions, the ones with stars, the 1', the 3', and the 4', that the hydrogen add to the wrong side of the DNA, and you get inverted configuration, and then the DNA is not okay. You added the hydrogen back, but now you have a structural change in the DNA that can cause problems with DNA synthesis. What else can happen? Radicals react with anything that's close by. That's the key thing to always remember. So a radical that's formed here can actually just jump around in the DNA and move from one sugar to the next sugar. If it's close enough to a hydrogen, it's going to do that. And the next situation is that you can get a strand break. The radical can move to the base. So then you have something here that we call a tandem lesion or a cluster lesion. And those are when you have 2 damaged spots or more in one helix. These are really bad. The cell can't handle those at all. And so they are highly mutagenic. What else can happen is that the strand can just start to fall apart to try to actually repair itself. And this gives you what we call prompt strand breaks. And those are also a big issue for the cell to deal with. Now, everything that we talked about over here, there's no oxygen around. So we know, of course, that cells, healthy cells, are highly oxygenated. So radicals love to react with oxygen. So if oxygen is there, that's the first thing that's going to happen. We'll form peroxyl radicals, and those will actually decompose to these types of substrates. When we look at those substrates, the first thing that becomes apparent is all of these different carbonyls. Carbonyl groups, of course, are electrophiles. The cell is full of nucleophiles. So creating these types of species then creates the situation for reactions that cause cross-linking. So that's an important aspect of what we're looking at. So we think about DNA structurally as B-form and duplex. Of course, when DNA is replicating, we see here that there are regions that are single-stranded, there are regions that are duplex, there are regions that are at junctions here where there's part duplex, part single-strand, there are proteins around. So one of the key issues that my laboratory is moving forward to is looking at radical generation in the presence actually of proteins. So we think that this is an important area. Of course, replication occurs in the S phase. And the S phase is when DNA is most susceptible to damage. So how do we study these questions? What are we interested in? So my laboratory wants to find out what the structure or what the chemical nature is of the small molecules that form when DNA is damaged by radicals. Why do we want to know this? There are two reasons why we want to know this. One of them is because then we have an idea of how things get repaired, how they're fixed, how we can actually avoid cancer. And it also gives us an idea if we want to create molecules that decrease the amount of damage or antioxidants, then we get to know an idea about the mechanisms that we're trying to target. So my laboratory specializes in synthetic organic chemistry, and we make molecules like this one, which are actually modified nucleotides. And what you see here in this situation, and this is the one that we're going to talk about right now, we have a ketone group attached at the position here where we want to form the radical. And this is the C3' position. So why do we put a ketone group there? What's the purpose of that? Well, ketones actually undergo a photochemical reaction when they're exposed to light at 320 nanometers that causes cleavage of the bond adjacent to the carbonyl, and this then generates a radical species and an acyl radical, and these then lose carbon monoxide and we get the radical of the sugar and an alkyl radical. So one of the things that you'll notice here is that I show cleavage so we don't have to worry about subsequent reactions occurring. So, in order to study this, we had to synthesize, of course, these modified nucleotides. And so we're lucky that we can start with commercially available thymidine or a nucleoside, and we just undergo a sequence of protecting group chemistry. So we protect here the 5' hydroxyl, we oxidize then the 3' hydroxyl with desmartin to get to the ketone. Then we form a protected cyanohydrin here, which is a mixture of isomers. We actually separate those, and then we get to this protected cyanohydrin here with the nitrile on the lower side of the molecule. To get to our ketone group, then we have to actually do a nucleophilic addition to the nitrile. That addition then actually gives us the imine that then undergoes hydrolysis, and then we get to our modified nucleoside with the carbonyl in place. Then we have to prepare this for oligonucleotide synthesis. We protect then the 5' hydroxyl with a dimethoxytrityl group, which is the common protecting group in nucleic acid chemistry, and then we actually convert this 3' hydroxyl to an H-phosphonate. So if you're familiar with nucleic acid chemistry, you know that usually phosphoramidites are used commercially for DNA synthesis. But because of the fact that we have this very bulky group here, we had to modify the chemistry in order to efficiently synthesize the oligonucleotides. So oligonucleotide synthesis is of course automated. We don't have to stand in the lab to do that. We punch the numbers and stuff into the computer and we walk away and we come back and our DNA is sitting there. And that's a beautiful thing for my students after they've done the multi-step synthesis. So what we do here is the control pour glass beads contain the unmodified oligonucleotide. Then we incorporate our substrate using covalent chloride as an activator. We use iodine, water, triethylamine, and pyridine to oxidize, and then we get our substrates here. So when we take it off of the synthesizer, it still has a protecting group here, and the bases are also protected. We cleave all of those things, take it off the bead, and then we have our modified DNA. Of course, then most of the time we have to do purification, which is usually done by reverse phase chromatography, and we analyze our substrates by multi-talc mass spectrometry. This is just a table to show you that we can do it in all kinds of substrates. We have the tertiary butyl ketone, we have the methyl ketone here, we even have the substrate that results from the radical trapping. We can make all of those things and we get very good coupling efficiencies at the modification. So how do we then study the products? So we take our substrates, then we expose them to light at 320 nanometers as a physiological buffer as we can find, usually some phosphate type buffer. We do it in the presence of 6 millimole of glutathione, and we use oxygen when we want to know what happens in the healthy cell. And then we actually analyze all of the products by ion exchange chromatography. So ion exchange chromatography gives us the ability to separate very complex mixtures to baseline resolution, and that's what you see here. So the interesting thing in this system that I'll tell you and then I'll move away from it is that a single radical here generates a lot of products. And we were the first group to actually show that the generation of one radical in an oligonucleotide can actually give you a lot of different products. And so this area of research under the aerobic conditions in oxygen has created a lot of work for us probably for the next decade to understand what's going on. But the next thing that we wanted to find out is what happens when we're actually looking at an unhealthy cell. So a very important aspect of understanding cancer metastasis and things like that is understanding the tumor microenvironment. And we know that the tumor microenvironment is different than the environment in a healthy cell. It's hypoxic. That is one of the key issues and one of the reasons why developing drugs to treat tumors is more difficult because we have no oxygen there. So we have hypoxic regions actually usually deep inside the tumors. The environment is acidic. It's usually in the range of pH 5 and below. They are nutrient deprived. And then they have variable amounts of reducing equivalents. And these are different reductants, with glutathione being the one that we focus on most often. So those are two very important differences between a healthy cell and an unhealthy cell. Why is it not moving? It's hypoxia, no oxygen, and glutathione levels that are highly increased or decreased. So we decided that we wanted to look at this mechanism to try to understand the radical reactions that occur here so that we may have a better idea of actually guiding the synthesis of radiation sensitizers and different molecules that can actually affect tumor treatment. So back in 1992, a proposed mechanism was developed in the laboratory of Jackie Barton, where she proposed that the radical, when it forms here, it undergoes an oxidation step, which then causes strand cleavage, and we get this ketone substrate, and then we actually get loss of the base here, so we're losing genetic information as well as getting a strand break, and then this decomposes to compound 8. And this molecule, to anyone who does MECChem know, that is a myco-acceptor, and a really bad one. So generation of this in a cell is a highly toxic event. So this is something that we really wanted to get involved in understanding. Over here is what occurs when we actually react the radical with glutathione. And glutathione, being a hydrogen trapping agent, will repair the radical, as I talked about before, or it will pseudo-repair it, as we like to call it, and give us an inversion of configuration at this position, which is a structural variation in the DNA. So if you remember my diagram I showed you on replication, DNA actually has many different structures as it's going through the process of replication. Some of it is single-stranded. There are points where it has a single-stranded region and an overhang region here, or the duplex region, which we refer to as overhang, and then some regions of duplex. So what we decided to do was synthesize oligomers and constructs to actually study the difference in the radical chemistry based on the structure of the DNA. So that's what we did here. So what you see on this slide is actually how we did this project. We took our modified oligonucleotide. We then actually annealed it to different substrates to make a single strand or an overhang or a duplex. We did the same photochemical transformation in the presence of glutathione, but in nitrogen, no oxygen, because we wanted to know what happens in the anaerobic part of the tumor. And what we actually found is very interesting. In single-stranded DNA, we see a large amount of actual reduction, meaning that the hydrogen atom can actually repair the radical, which was very interesting to us because we thought that probably we would see more strand breaks there, and we'll talk about why that is in a minute. And we see actually 22% strand break here and 88.5% here. When we get to the overhang, then of course we have single-stranded and duplex. We see a slight drop in the amount of reduction as well as a drop in the amount of strand break. When we get to the duplex, we actually see quite a bit less reduction and less strand break. And it's interesting to us now, you'll notice that the amount of material recovered goes down. And so we actually believe that we have alternative mechanisms occurring here that we're not being able to identify. So we're trying to actually figure out why these numbers are going down. So what you'll notice here is that the precursor, which you can't see, is in this T area, where it's only T's. When you go down here, we actually put the precursor in an area with other bases. And then you see that the chemistry actually changes quite a bit. We see much less reduction and strand break, and our mass balance is even lower. So that gives us an indication that some of the other bases may be interfering with the radical chemistry. The other interesting point that we made, this is actually very significant in this field, is that when we do these types of reactions, this reduction reaction, we can actually measure the amount of real repair to pseudo-repair. And what we found is in single-stranded DNA and in overhangs, we almost get a 1-to-1 ratio. So it's completely non-discriminate. But if you notice in the duplex, which is here and here, we get much more of the repaired substrate. So that gives us an idea that what we're seeing is a template effect. By simply forming the duplex, the DNA is then more protected from actual damage. And so that's probably one of the reasons why the DNA is usually in a duplex form, because it protects it from damage. So that was a very important aspect. And radical creation at other sites also does the same thing in duplex DNA. So that gives us a very good idea as to that. So basically here, this is kind of a summary. You can see all of the different kinds of products that come from the 3' radical. Here are some of the electrophilic species that we see here, the ketone and the 2 aldehydes. This is the inversion of configuration. The ketone is in both pathways. So why is this important in cancer etiology? So if you see here, this is the aldehyde that we just showed that forms from the aerobic pathway. It's known that this compound, or this oligo, degrades to glyoxyl. Glyoxyl then reacts with deoxyguanosine and forms this adduct. This adduct is highly mutagenic and has already been shown to be carcinogenic as well. At the bottom here, you see this substrate. Now this substrate comes from the 5'-aldehyde, and it actually reacts with the adenosine base, gives an abasic site, which I told you is also mutagenic, and gives us this compound kinetine. Now kinetine actually as a compound has antioxidant properties. So that's a very interesting thing to see, that we create through DNA damage a molecule that is actually antioxidant in nature. So basically here, this is just a conclusion that tells you what I said, and one of the most important things is that the duplex DNA facilitates the proper chemical repair of the radical. We were able to do all of these things, and we were the first to show the formation of this deoxyxylose thymidine. So the next step here is to actually study the effect of that substrate on DNA synthesis. So the next topic I'm going to talk about also has to do with radicals, has to do with sugar radicals, but from a different source. And these radicals come from what we call LEEs, or low energy electrons. So whenever I talk about this topic, people always ask me, well, when are we exposed to low energy electrons? So I'm going to start off by telling you that. So one of the most important things to remember is chemotherapy radiation therapy is used to treat cancer patients. So when they are exposed to radiation and their tumors are treated, then the incident radiation is usually high at first, but as the particles actually enter into the tumor, they lose energy. And that is what creates then a large amount of low energy electrons. So actually what we're looking at then is the actual mechanism by which we treat also can have further effects by the creation of other species. So that's what we're going to talk about a little bit here. We'll just go through a few things here. Of course, we know that radiation interacts with DNA in living cells. And we pretty much know that this interaction with DNA is one of the specific reasons why radiation causes cell death. So that's a very important thing. It's useful in the radiotherapy to kill cancer cells, but it also initiates the formation of cancer cells. So that's always an issue that we have to deal with. Radiation particles produce radicals, ions, and free electrons, all of these being highly reactive species. And a high percentage of these free electrons are low in energy. And when we talk about low energy, we're talking about 0 to 20 electron volts. So some work that was done in Canada in early 2000s showed that when the DNA is exposed to electrons between 3 and 20 electron volts, you get a peak of strand breaks at about 10 eV. This was the first time that this was observed because the popular belief in the radiation oncology field was low energy electrons had no effect on the cell. And so when this paper came out in Science in 2000, it caused a very big kind of issue in the radiation oncology community. So this actually just shows how the DNA breaks. It breaks at every site at the phosphate groups. And what you see here are some of the substrates that form. We end up with modified oligonucleotide strands, and we end up with base release. And you see here the modified fragments. That's what we're interested in. We want to know what actually happens to the DNA. So also the same group, they've been very, very instrumental in explaining how these things happen, but they actually came to me and said, we don't know how to figure out what happens to these radicals when they form. Can you look at it? And so that's why we started this project. And you'll notice here that one of the radicals is at the 3' position, but it's missing, of course, the rest of the strand because this radical, the electron actually adds to the phosphate group from the base to the phosphate. This causes a high energy situation, and the strand breaks and you get this radical in this one. So we're interested in finding out what happens to that radical. So again, we use our same approach. We use radical precursors. Here you see the same tertiary butyl ketone group. Over here, we actually use also a different functional group this time, and that's the phenylselenide group. So again, we have to synthesize these substrates and learn to study their chemistries. We start with this dimethoxytrilo-protected mesylate. We can convert that very easily to an anhydrothymidine. It then is exposed to the nucleophilic phenylselenide, and we create the substrate that we want here with the dimethoxytrilo group on the 5' position. And then we just deprotect it, and that gives us our modified nucleoside. So at this position, of course, we can have two different isomers, and we know that sometimes the chemistry can be very different. So we actually synthesized both substrates. We made then the mesylate again, did not convert it to the anhydro, just treated it with the phenylselenide anion, and that gives us the other isomer. And then we deprotect it, and we have our substrate. We also synthesized both isomers of the tertiary butyl ketone group. This one started simply with actually a tertiary butyl dimethylsiloprotectidimidine, and this has a hydroxymethyl group here. Actually, the synthesis of this is probably about a 7-step synthesis to this molecule, and then we have to go here. We do an oxidation to the aldehyde. We alkylate to get our tertiary butyl alcohol, oxidize again to get to the tertiary butyl ketone. The other isomer is a little bit more difficult because of the stereochemistry. Here we start with a Barton ester. We actually perform a reduction here, a reductive addition. We put on a styrene moiety. We do an oxidative cleavage to get to the aldehyde. The aldehyde is then alkylated in the same fashion as the previous substrate. Then we oxidize and we get to, and deprotect, and we get to our compound. So the first thing that we always have to do when we create new radical precursors is determine, does the substrate actually make the radical? That's the first thing we do. So we took these substrates as monomers, we exposed them to light, only in the presence of tributyl tin hydride, which is also a strong hydrogen atom donor. And as you can see from the reverse phase chromatography here, our photolysate, which is our reaction mixture, gives us only the reduction product here. These are our controls, that's our starting material, and these are our standards. So mentioning standards here, I should say that every molecule that we identify, we independently synthesize. Because we don't want to just rely on mass spec data or things like that, so we independently synthesize every product that we find, which creates a lot of work for my students, but they learn how to do synthesis. So, you'll also see the same thing occurs in the next two slides. So the tertiary butyl ketone actually gives us the same thing, but this one is actually less efficient in this case because it takes a lot longer to convert to the actual product. So we decided to pursue then the selenide chemistry, and we did actually make then the H-phosphonate derivatives from this. And we had to use slightly different chemistry, the substrates were different. So we used this phosphorylating agent here, and we actually could form then our H-phosphonates in relatively good yield, not as good as we would like, but they're okay. And then we incorporated all of these substrates. These are actually products that we expected from the photolysis. We synthesized oligomers that contain all of these, and what you see here are the theoretical and experimental mass values using multitop, and we were able to obtain those in very good purity. So when we actually did the photolysis of the oligomers, we got this mixture of compounds. One of these is then the 2,3 unsaturated nucleoside. This is the 3,4 unsaturated. This is thymidine, and this is a phenyl derivatized substrate. These are not the products that we thought they would be. They actually gave us a completely different distribution. And knowing the chemistry of phenylselenides, we actually determined that the phenylselenide radical, I said we're always making two radicals. The phenylselenide radical doesn't just go away. It does all kinds of other chemistry. So through these experiments, we determined that this is definitely not the ideal precursor. However, we're making this compound here, 14E, in very high yield, and it is actually a damaged product in another pathway. So we can use it as a tool to actually study DNA damage involving this substrate. So in the future, we will probably go to looking at our tertiary butyl ketone derivatives to do this work. So in conclusion here, we can make these modified nucleosides very successfully, and they represent important tools for the identification of low energy electron damage. They've been successfully used, but they generate products that we don't really want. So we still have a lot of work to do in this area, and this is one of the areas of current focus in my laboratory. So the students who actually did the work, the aerobic work was done by Rehana Zaidi. She's now a laboratory chemist at a pharmaceutical company in Alabama. This is Suat. You have seen her running around here. She is now professor at Just. This is Ruthena. That's the other sister. She's also a professor there. This is my student, Fernand Beatty. He is from the Ivory Coast, and he's working on derivatives. He has to do the synthesis of some of the products we identified. And this is Nicholas Amato. He did all of the work with the different substrates that we look at for damage. And I'd be very happy to take any questions.
Questions?
Yes.
Dear Amanda, I want to ask you, so right now you have mentioned that the mutation will be in the 3' position. And as we know that the replication will occur in the orientation of 5' to 3'.
Yes.
And since the mutation will occur at the 3' position, what kind of mutation will happen? Is it a nonsense mutation, or is it a frame shift, or what kind?
That's the key issue. Basically, we know that by forming the 3' radical, we get a strand break. And so we know if we get a strand break, then most likely it's going to cause the replication fork to collapse because of that fact. So the kind of mutations that we expect are the ones that are usually associated with that kind of replication fork failure. So exactly what kind we don't know yet, because we have to actually work with other people who are doing those kinds of assays to determine, but we expect it to probably be a frame shift type of mutation.
Something else. I have noticed in one of your structures that there is an alteration in the binding mechanism. So there is a phosphatic group will be linked to the oxygen species, which is located on the upper location. So this one will cause a shift, but did you examine what's the effect on the DNA polymerase or something like that, or not yet?
Not yet, and that's also interesting because we don't do molecular biology or anything like that, so we're looking for collaborators. So if you want to collaborate, let me know. But we did look at the structural impact. So we know that when we get the phosphate group over on the upper face, it causes incredible instability in the duplex. So we know then that it could be a spot where we will actually see bubble formation or things like that, because it seems that the DNA is actually breathing at that location. So the stability goes down quite a bit. We've even looked at that energetically, and it has a huge impact. So it's an important question. We do know from the 4' position, when you invert configuration there, you get complete polymerase stoppage at that position. It can't go past that. So if this is any indication of what we're going to see, it probably has that kind of impact.
Okay, and something else. Okay, you have mentioned that there is the major groove and the minor groove. Okay, in the major groove, the carbon number 5, 4, and 3, it's outward, and 1 and 2, it's inside.
Yes.
Okay, so I have noticed that you have mentioned at C2, there is an active species or electron that are attacking that position. So I don't understand how it would be attacked when they are buried inside, not outside.
Well, this is the key issue, that the DNA is not always duplex. So usually we see very low amounts of products that are related to the 2' radical formation. However, we see a lot of products associated with 1' radical formation. And that most likely has to do with the fact that the base is there. So the base actually typically attracts radicals, so we do see a discrepancy in that. But there are some calculations that have been done to determine which hydrogen atoms are energetically going to be abstracted first and which ones are more accessible. The 5' are the first ones to go, it seems, because they're more accessible. Energetically is the 1'.
Okay, thank you.
Yeah.
Last question.
My name is Anam Shahada. Thank you, Amanda, for your lesson. I would like to ask you how a person can prevent him or herself from having a cancer.
Oh, if I could answer that question, I'd be the wealthiest woman in the world.
Yeah.
One thing that is important, and there are several things from my research and what I understand. So trying to avoid situations of chronic inflammation is going to be very important as we do more research. We're going to find more situations where inflammation contributes to cancer development. Another area that I forgot to mention is esophageal cancers that come from smoking. Those cancers are also not just because of the benzopyrines and carcinogens that collect in the esophagus, but also because of chronic inflammation. On the other side of that, when you have acid reflux disease, which comes from eating a very high-fat diet, that can also cause chronic inflammation in a lower area. So that's one of the key things. So we have to be careful. When they say eat a good diet, it's true, because that's going to give you the nutrients you need, the antioxidants that you need. Antioxidants are definitely very important in trying to prevent cancer. The other thing, because I'm a chemical toxicologist, is you should avoid toxins. If you can tell me how to do that, I would be very happy, but pollution is awful for you, and things like that. So you have to be careful about those kinds of things. But of course, some cancers are genetic in origin, particularly breast cancer. You can't do anything about that. So I don't think we'll ever be able to say, this is how we prevent cancer, because it's so many different diseases.
Okay, thank you so much.
Very good question, thank you.
We have to give just one exception for Dr. Layla.
Thank you very much for your excellent lecture.
Thank you.
Now, my question, you said at the end that radiation causes
Cancer.
cancer.
Yes.
So, this means there should be a change in the way of treatment, because so what do you comment on that? I mean, this is very important discovery or conclusion out of your research.
That's a difficult question. There is nothing better to treat most solid tumors than radiation. Okay, so we really probably won't for quite a while be able to avoid that. There are other therapies like photodynamic therapy, which uses dyes and you can use light, and that has a lesser incidence of causing secondary cancers. And that's usually the issue. The radiation that you use to treat one cancer will cause a secondary cancer. Okay, but I think chemotherapeutic agents, of course, we need to keep working on that as hard as we can to see if we can find ways to treat solid tumors. And I wish I could answer that question better, but I don't think radiation is ever going to go away. I don't. Women who have the genetic marker for breast cancer get mammograms at a lesser amount than women who don't, because they know that it has also the ability to cause cancer. So that's part of the issue, yeah. So we have to be a little careful about those things. Good question.
Okay, Amanda, thank you for the nice and interesting presentation. Unfortunately, we're running behind schedule, so we cannot take more questions. Probably whoever have any question can ask Amanda after the session. Now we are again pleased to welcome Dr. Yusuf Sari from the University of Toledo. Dr. Sari is an assistant professor, and he got the Young Scientific Award, Sigma Phi Award in 2011. As well, he got this Research Scholarship Award in 2012. Dr. Sari received his PhD in neuroscience from here and Marie Curie University, and he did the postdoc training and the research and was a research faculty at Indiana University. And now he's an assistant professor, as I mentioned, at the University of Toledo. His talk will be about the glutamate transporter 1 as a potential therapeutic target for the treatment of drug abuse and neurodegenerative diseases.
Assalamu alaikum. I'm going to switch gears to pharmacology. But we really do appreciate the chemists, how much they provide to us to test those chemicals in the stage of pharmacology. I really did like your roadmap where you show from synthesis, screening, and I'm the guy who's working with my mice and rats. So that's all I do. I test compound for treating some of the disease here. One of them is drug abuse, including alcohol or cocaine. And the other one is neurodegenerative disease, so the one we're going to be talking about is Huntington's disease. Okay? So what I'm going to do, I'm going to talk about kind of briefly. This is my plan. It's introduction. I'll talk about the role of one of the compound that is targeting glutamate transporter 1 and Huntington's disease. Then we talk a little bit about cocaine, ceftriaxone as a target, kind of targeting glutamate transporter to attenuate relapse to cocaine behavior. Then the last part, briefly, I will talk about the compound I'm using to target the glutamate transporter in alcohol preferring mode. Okay, so really the glutamate transporter is located right here, so we can see in this glia. So, for example, you have communication, neural communication with glia. When you think, when you learn, basically it's neural communicating with the other. Like I'm talking right now, I have my neuron that can involve in the process. But glia, what they do, they support this guy, they support this neuron. It's like a housekeeper. And at this point, what they do, to focus on my talk, is this glutamate transporter that regulates glutamate uptake. Okay? If you have a lot of glutamate in the synapse, this glutamate transporter will regulate them. So they kind of uptake those glutamates inside glia. Okay? And this particularly, we have, of course, we have many glutamate transporter, and this one is regulate 95% of glutamate. And here is actually the neurocircuitry of drug abuse, including alcohol, cocaine. And you can see you have here prefrontal cortex. So if I for example, I cut my brain like this, what you're going to see prefrontal cortex. And in the depth, you're going to see you have some area here called nucleus accumbens, ventral tegmental area. This is what we call a neurocircuitry, a very simple neurocircuitry of drug abuse. You have prefrontal cortex, which is a key for, how we call it, making decision. Okay? So remember that, making decision. And you have these glutamatergic projections, okay? Then what they do, they release glutamate again. So for example, an addicted person, what's going to happen when I provide the addicted person with alcohol, cocaine, you're going to have a lot of glutamate floating here. That's going to induce dopamine, which is basically a center of reward. Well, it's a neurotransmitter for drug reward or drug abuse in general. Okay, so you see that when you see alcohol, when you hear it, this is what's happening. I'm gonna keep going. Neurocircuitry drug abuse in general is very complex. I'll make it very simple. You can include amygdala, which is involved in anxiety. Okay, and this amygdala is also projecting to this area, including the cortex, ventral tegmental area, nucleus accumbens, and they are, of course, glutamatergic projections. Okay? Now I'm going to give you a briefly what's going on with drug abuse, neurocircuitry, and since I will be talking about Huntington's disease, I'm just going to briefly talk about why I'm interested in glutamate control. So here, this is a very complex. I don't want you to kind of go deep. We have to spend a lot of time to dissect piece by piece. But what I want you to see, this is neuron, okay? And this is glia. And this is my glutamate transporter here. This fragment here called mutant Huntington protein. When it's in general Huntington from what it is. Like you and me, we have a CG repeat about 25. When that CG repeat exceeds 25, then you're going to produce what you call a mutant Huntington protein. So more than 25, sometimes, the scientists, they say more than 4, etc. So when you have that protein expressed, that what's going to do is going to affect the whole machinery, including vesicular transport, including transcription, etc. But one of the problem with Huntington's disease, you have a lot of glutamate protein. Too much glutamate, that's going to kill the cells. And because with this mutant, it's interfering with the transcription of that glutamate transport. Okay? So I want to do, I have my compound to increase that the glutamate transport that it was reduced in Huntington's disease. Okay, you have idea. Now, this is what the hypothesis. The hypothesis is to find a compound targeting our glutamate transporter, okay, which is regulating 95% of glutamate. And glutamate is exotoxic. When you have a lot of glutamate, it's going to kill the cells. And that's what you can see for like ischemia. When a person, goes to ischemic stage or have like accident, what's going to happen is a lot of glutamate. Amyotrophic lateral sclerosis, you have a lot of glutamate. So Huntington's disease, we talk about a lot of glutamate. Now here is what's happening. When you have a lot of glutamate, this is coming from for example, neuron, going to be released, okay? It's going to be floated. Here what it's going to do is going to stimulate this gas, okay? Either it's going to lead you to go drink alcohol or cocaine, or if there is excess of glutamate, then it's going to kill the cells. And it's going to induce what we call oxidative stress, okay, free radical. Now consider we have a compound that's we're going to deliver the compound, that's what's going to happen, it's going to overexpress this glutamate transport. What's going to happen? What do you think is going to happen? You can't answer. You're going to have less glutamate protein in the cells. Okay? So you have less glutamate, this is a good thing, so you're going to have less stimulation, less craving, less toxicity, etc. Okay? So this is how we start. We were looking for a compound. We have a system, our target system, which is glutamate transporter, and this is where we started our journey. By this group at Johns Hopkins, when they discovered many compounds, including cephalosporin, ceftriaxone maybe, what they showed that the compound is basically can upregulate the glutamate transporter, and it attenuates, actually reduce ELS, improve the progression of the amyotrophic lateral sclerosis. Okay. Now, when we saw this paper, we say this is a good compound that's going to target all protein, and it's not going to be working on our cocaine. It's going to be working on cocaine. This is the hypothesis at the beginning. It's going to work on cocaine. We have actually at the time we have the cocaine syrup, a model of cocaine, alcohol, Huntington's disease, and this is actually the cephalosporin. You're going to say because the pharmacies, probably you are all, most of you know cephalosporin. You can say what's antibiotic and how this antibiotic can increase glutamate transport. Well, by accident. The good news, the compound crosses the brain barrier. It not only can treat what the cephalosporin can do, it can treat meningitis. Because meningitis is in the brain, okay? So the compound is going to go and treat meningitis. But by accident, it does affect the transport, retinal transport. It's neuroprotective. So I'm going to keep going. And this is actually the group, what they did, they tested ceftriaxone in ELS model, which is you have laroglutamine that's killing the cells. And right now, the ceftriaxone, which is beta-lactam antibiotic, it's in clinical trial phase 3 for 2